Hey everyone and welcome. Today we are diving into one of the most game-changing techniques in all of modern biology, polymerase chain reaction, or PCR. Now, you can think of it like the ultimate molecular photocopier, a tool that has totally revolutionized how we study the very building blocks of life.
But let's start with the basic problem it solves. Picture this. You're a scientist, and you've got this tiny, almost invisible sample of DNA. Maybe it's from a crime scene, a fossil, or a patient's blood test. The amount is so small, it's basically impossible to work with. So how on earth do you analyze something you literally can't even see?
Well, the answer is both super simple and kind of mind-blowing. You don't. You don't try to study that one single molecule. Instead, you make billions of identical copies of it. So how in the world is that even possible? Let's get into it.
You see, the biggest challenge for scientists has always been about quantity. Starting with just one or a few strands of DNA is like trying to read a single word written on a single page in an entire massive library. To find it, to understand it, you need to amplify that signal. You need to make that one little word shout.
And that's precisely what PCR does. At its core, PCR is all about amplification. It hones in on a specific segment of DNA that you're interested in, and then it just replicates it over and over and over again until you've got a massive, detectable amount from a nearly invisible starting point.
So, how does this molecular photocopier actually work? Honestly, you can think of it a lot like baking. You need the right ingredients, you need to mix them together, and you need an oven that can hit some very specific temperatures. So let's take a look at the recipe for making billions of DNA copies.
Okay, so our recipe has 6 key components. First, you need the template DNA. That's our original blueprint. Then you need primers, which tell us where to start copying. Of course, you need the DNA polymerase, the enzyme that does all the work. And that enzyme needs building materials. Those are the nucleotides. To make sure everything works smoothly, you add a buffer. And finally, you stick the whole concoction into the thermal cycler. That's our high-tech oven.
So let's start with the template DNA. This is your master blueprint. It could be from a single drop of blood, a hair follicle, or a swab from a patient. This is the precious original copy that holds the genetic story we want to read.
Next up, we have the primers. These things are so cool. They're like tiny programmable GPS coordinates. Scientists design these short DNA fragments in a lab to match the exact start and end points of the gene or sequence they're looking for. This is super important because it makes sure we only copy the specific part we want to study, and nothing else.
And then we have the star of the show, DNA polymerase. This is the master builder. It's an enzyme that zooms along the DNA template, grabbing those free nucleotides and stitching together a brand new identical strand. Now, the polymerase used in PCR is really special. It's called Taq polymerase, and it was found in bacteria living in hot springs, Thermus aquaticus. This means it can withstand incredibly high temperatures without falling apart, which, as you'll see, is absolutely crucial.
And finally, we have the hardware, the thermal cycler. It's basically a really sophisticated, programmable oven that can change its temperature with insane speed and precision. This machine automates the whole thing, cycling through different temperatures over and over to drive each step of the reaction.
So you've got all the ingredients in the tube. How do they actually come together? Well, the magic of PCR happens in a really simple, repeating, 3-step cycle that's driven entirely by temperature. You can just think of it as melt, stick, and copy. All right, let's break down those 3 steps. They're called denaturation, annealing, and extension. And each one is kicked off by a very specific temperature change inside that thermal cycler. Okay, so the cycle starts by cranking the heat way up to about 95 degrees Celsius. This is hot enough to literally melt the DNA double helix apart into 2 single strands. Then the machine rapidly cools down to about 55 degrees. This lower temperature allows our little GPS primers to find their targets and stick on. And finally, the temperature gets bumped up to 72 degrees. The perfect sweet spot for our Taq polymerase to get to work copying, building a brand new DNA strand from each of the original templates.
And this is where the real power comes in. After one cycle, our one DNA molecule has become 2. After the next cycle, we have 4, then 8, then 16, 32, and so on. The machine repeats this simple cycle about 30 times, doubling the amount of our target DNA with every single pass. It's exponential growth in a test tube.
And the result?
After about 30 of these cycles, which only takes a couple of hours, that single invisible DNA molecule has been amplified into over 1 billion copies. Suddenly, you have more than enough to see, to analyze, and to work with.
Now, this is way more than just a cool trick for the lab. It's a tool that has fundamentally changed our world. So let's talk about why this molecular photocopier matters to you and how it jumped from the lab right into our everyday lives.
In forensics, PCR is an absolute cornerstone. You know how on TV they find a single hair or a microscopic drop of blood? That tiny sample has enough DNA to be amplified with PCR. Scientists can then generate a unique DNA fingerprint, linking a suspect to a scene with incredible accuracy. It's completely changed criminal justice. In medicine, PCR is a frontline diagnostic tool. Instead of waiting for your body to create antibodies to an infection, doctors can use PCR to look directly for the virus's own genetic material. This means they can detect diseases like HIV, hepatitis, and countless others much earlier and with amazing sensitivity.
And get this, it even lets us look back in time. Scientists can take tiny, degraded fragments of DNA from things like fossils, mummies, or ancient skeletons and use PCR to amplify them. This has opened up a window into the genetics of extinct species like the woolly mammoth and even helps us understand our own ancient human ancestors. It's just incredible.
So, from this simple idea of just copying DNA, we have gained this incredible power. The power to solve crimes, to diagnose deadly diseases, and even to rewrite history. PCR is the key that lets us unlock and read the amazing stories hidden deep inside the code of life. It really just leaves you wondering, what story will we uncover next?